Welcome to Almost Here, Round the Corner Future Technology podcast with Richard Jacobs. Future technologies poised to transform our lives for better or worse are the focus of this podcast. Almost here means these technologies are now here and starting to be used, or just around the corner, from Bitcoin to artificial intelligence, 3D printing, blockchain, virtual reality, and more.
Hello, this is Richard Jacobs with the Future Tech Podcast. My guest today is Jagannath Padmanabhan. He's a PhD and a postdoctoral research fellow in Dr. Jeffrey Gertner's laboratory in the Department of Surgery at Stanford University. He's a bioengineer by training, PhD from Yale University in 2016. And we've been talking about his research interests, which deal with the interface of bioengineering, surgery, and data science, and looking at the role of mechanotransduction in biomedical implant rejection, which we'll get into. So, Jagan, how are you doing today?
I'm doing well. Thank you, Richard. Thanks for having me on the show. And before we start, I would like to say that I've listened to many episodes of the podcast series, and I've really enjoyed them. Very informative. So congratulations to you and your team for putting together a good show.
Oh, thank you. Yeah, no problem. So tell me about your current research. What is your focus right now? And let's boil it down to a simple version of it, and then we'll start getting into questions about it.
Yeah, I'd be happy to. As you just said, I work as a postdoctoral research fellow in plastic surgery here at Stanford. So my research interests are basically focused on biomaterial tissue interactions and novel biomaterial development. As you said, my previous graduate studies have been at Cornell and Yale University. So for the past almost 10 years now, I've been doing interdisciplinary biomedical research focused on how, our body reacts to biomedical devices, and especially in particular, the role of mechanical forces in how the biomedical device rejection happens.
So what happens right now? What kind of biomedical devices, like pacemakers or artificial?
Yeah, that's a good question. So, as there's a whole variety of biomedical devices that we use in surgery today, taking from pacemakers in cardiovascular medicine to breast implants in plastic surgery to catheters for chemotherapy and so on. What I do is I started at Stanford about 2 years ago, and I started this multidisciplinary collaboration with different departments within surgery. So with plastic surgery, cardiovascular medicine, pediatric surgery, and neurosurgery to collect different kinds of implants that are used in patients. And when they're taken out due to a variety of reasons, I collaborate with the surgeons to get those biomedical implants. And I study, what kind of cells and proteins are there on the implant using some of the new technologies that are out there. For example, single cell sequencing technology. So we get an idea of what kind of cells and what kind of proteins are involved in the biomedical device rejection process. And that gives us new targets to go after when we think of therapies to increase the lifespan of these devices. As you might know, every artificial material known to man is sooner or later rejected by the human body. So we want to understand how this rejection process happens and how we want to evade the rejection process.
So there's no implant that anyone's had that they accepted long term. They always reject it over a period of time.
There's actually so the way the rejection process happens is this process called foreign body reaction. It's literally what it sounds like. Whenever you introduce a foreign body into the human body reacts in a certain way. It deposits certain proteins. There are certain inflammatory cells. Eventually, there comes fibroblasts, which produce a lot of collagen. And this collagenous, it forms like a capsule around the implant. And once that is the body's way of, isolating that foreign body from the rest of your body. And this is the body's defense mechanism. But when it comes to biomedical implants, this is not such a good thing because we want the biomedical implant to, not be isolated from the body and interact with the body. There are certain kinds of implants that would last longer than others, but basically all the artificial materials that we know elicit this foreign body reaction to a greater or lesser extent.
Well, what happens over time? Does a layer of material build up over an implant to where it becomes non-functional, or how does it work?
Yeah, so it works differently in different implants. For example, in breast implants, which is the largest number of implants in plastic surgery, one of the things that happens is when the capsule layer forms, it can actually contract the implant itself. So, the breast implant can get deformed, it can cause pain, and it'll basically stop functioning. So there's mechanical failure of the implant. In other things, like for example, the new biomedical implants that everyone's working towards are these biosensors. For example, in vivo continuous glucose monitors, and these implants, the foreign body reaction is even more of a problem because even a small capsule layer that forms is going to change the readings of the blood glucose that you're trying to read, right? Because once there's a capsule, there's less amount of interstitial fluid or blood that can get to the implant, and hence your readings go down. In fact, the average lifespan would be, a month or 2 months for the commercially available in vivo sensors. So we are able to figure out a way how to stop this foreign body reaction or how to stop this rejection process and increase the lifespan of these devices. That would be a tremendous help for a lot of patients, millions of them in the US alone.
So what's the pathway you think that would stop the rejection or slow it down to where it be for a lot longer?
Yeah, that's a good question. There's a few different ways we can get at it, which is why my research interests lie at this intersection of bioengineering, surgery, and data science. One of the things we can do is, and one of the things we've been understanding more and more now, is the role of mechanical force in biomedical implant rejection. Dr. Jeff Gertner, who is my advisor here at Stanford, had shown that mechanical force plays a big role in wound healing, which is a very related process. Because when you implant a biomedical device, you're essentially creating a wound and then placing a device inside of that wound. And what we've realized is when there is an increase in mechanical force, there is fibrotic processes that get activated and inflammatory processes that get activated. And the same things happen in biomedical device rejection. And so that would be one area to focus on. Like one is to understand how mechanical force accelerates device rejection, and also how we can come up with therapies to stop it. The other angle is to really understand what's going on with the cells, because obviously we've studied foreign body reaction for many, many years, and there's some statistic that says only 2/3 of the research at the basic science level ever gets translated to the clinical level. And even those haven't found too much success. And that's because to increase the lifespan beyond what we have currently. And that's because the identity of these cells that we know of, for example, we know that macrophages and fibroblasts are involved in this foreign body reaction. But now with newer technologies like single cell sequencing and such, and data science technologies, what we're realizing is that these cells that we talk about, like fibroblasts and macrophages, these aren't really one cell type. There's actually multiple cell types within them, transcriptionally active subtypes of cells, one subgroup of cells which could really be doing 90% of the damage. So now with these data science techniques, we're able to isolate, okay, which is the subgroup of cells that's really causing the damage, and then we can do some drug delivery-based techniques to target that specific subset of cells. And this is all on the biological side. During my grad school work, we worked on some. Please.
One thing. We're jumping around to a lot of topics. I just want to go a little bit slower.
Sure.
You said that mechanical movement of an implant itself causes an immune response or a scarring response. Like,
Correct.
Have you looked
Correct.
at when people have a bone spur or something mechanical, but native to their body on the inside, torn cartilage, whatever it is, that creates some kind of mechanical stress, and how the body reacts to that? Have you looked there for clues as to why an implant would cause, similar effects?
Yeah, maybe I should start with what happens in wound healing, and that'll give us a nice segue into what happens during implants, because it's a sort of newer idea, and there's an animal model that I'm developing. So in wound healing, what happens is that, let's say there is a certain wound that is created due to, let's say, a surgical scar or some kind of trauma, an accident or something, right? So when the wound heals, when people started studying them in mice, mouse wounds healed, much better than human wounds. And for a long time, we didn't understand why that happened. Later, people realized that mouse skin has certain different properties. There's an extra muscle layer, but it all came down to the fact that when you create a wound in a mouse and the wound heals, the forces that the mouse skin is experiencing is very different from the forces that the human skin and human environment is experiencing. As you know, the skin itself is under tension all the time. And when people did these experiments where in mice you would create a wound, and as the wound is healing, you would apply, human levels of force, you would create a human-like scar formation. So what I'm doing currently is developing a new animal model because mouse is obviously the preferred model to study any disease model, including biomedical device processes. And what we realized is that the forces that a biomedical device faces in the subcutaneous pocket in a mouse is very different from the forces that a device faces in humans. And now we've developed a way to, apply extra forces in the mouse, and then the reaction that the device gets in the mouse is more similar to what happens in the human. So this is an indirect way of getting at the fact that, the application of high mechanical forces, high mechanical environment around the biomedical implant is part of the reason of why these inflammatory and fibrotic process or the scar forming process really gets activated.
Okay. So what does that tell you to do with implants? Do they have to be a certain shape, or there's really nothing you can do? It's just, they're they're taking the position of something that it was either there or not there, and you're going to have this kind of reaction no matter what you do?
No, there's a few different things we can do. For example, one thing that we started, for example, my grad school work, what we were doing is that we started manipulating the surface of these implants. So we created nano biomaterials, so biomaterials with surface nanoarchitecture. So imagine a cell sitting on a flat and smooth surface versus a cell sitting on a bed of nails made of, nanoscale architecture. So obviously the cell would behave very differently when it's sitting on a bed of nails versus sitting on a smooth, flat surface. And this is basically a tool that we can use to manipulate the cell behavior locally. So the way the cell reacts to mechanical forces and other factors can be changed by changing the material surface. So that is one strategy. And the other strategy is, now that we realize that it's the mechanical forces that are making the device work differently, or the rejection process have accelerated, what we can do is look at what we call mechanotransduction molecules. These are basically, genes and proteins in the cells which are sensitive to mechanical forces. For example, in wound healing, we've looked at focal adhesion kinase. It's a protein that is involved that transduces mechanical signals into fibrotic processes in fibroblasts. And then if we target these mechanotransduction pathways, then we're basically cutting off the signaling between the mechanical force and the end result of scar production. So we want to take that kind of an approach with biomedical implants too. Once we prove that it is the mechanical force, which we have quite a bit of evidence now, that's making the accelerated rejection process happen, if we can now target drugs that can inhibit or attenuate the mechanotransduction process in the local environment, then that's the way we can get around the process.
How long, what's considered fast rejection versus normal rejection? How long, I'm sure it depends on the implant, but on average, for a pacemaker or other types of implants, how long does it take for rejection to happen if it's normal versus accelerated?
Yeah, that's a good question. And there is a lot of sort of variability in the studies too. And that's another reason why it's complicated. So let's start with biosensors. That's the most sort of acute kind of rejection. Again, as I said, the commercial in vivo continuous, glucose monitoring devices, the average lifetime is 2 weeks to, let's say, maximum of 2 months or so. With breast implants, it can last up to 7 years. With pacemakers, again, there's a lot of variability. Technically, the battery might last until 15 years. But let's say you have to change the battery. The other problem is because of the foreign body reaction, when you have to remove the implant is stuck to all the scar tissue that has formed around it. So removal of the implant itself poses some risks that are associated with the process. So the same implant in different patients also leads to different kind of reactions, which we don't understand why that happens. Again, there's a lot of variability there as well. So there is no fixed timeline for every device. Obviously, there are average lifespans, and depending on, if it's a passive implant, that is a mechanical support, that lifetime is a little bit better. But if it's an implant like an active implant, like a biosensor, the lifetime is very, very low. And obviously, regardless of the actual lifetime, our goal is to be able to increase the lifespan of these devices across the board.
Got it. What have you discovered with altering the surface material of implants? What seems to prolong the rejection, and why is your guess?
Yeah. So what we've done is, again, I've looked at a few different things. So the one thing I've done is collected these implants from, different departments of surgery. So I've collected pacemakers, breast implants, chest batteries that are used for deep brain stimulation, and so on. And looking at these cells, we are arriving at certain kinds of mechanotransduction pathways that are upregulated, specific pathways. So that is one sort of way we can go down to figuring this out. Another evidence is that in mice, we are able to recreate, sort of better recapitulate what happens in humans when we apply a lot of mechanical force, human levels of mechanical force in the mouse implant. So all this evidence is pointing towards the fact that mechanical stimulation of these implants is accelerating the rejection process. And what we are trying to do is, one, understand how that happens. Second, identify the specific subgroups of cells and mechanotransduction pathways that are involved in this process. And the second step, which we are yet to get to, is basically devising drug delivery therapies that would target these specific pathways and so on.
Well, do you find, again, to refer to an earlier question, when do you find examples in the body of naturally occurring stuff like a bone spur or, again, torn cartilage that simulate what happens to an implanted device? Is there anything in our body that happens naturally or unnaturally that causes this mechanical wear and tear, that causes scarification or fibroblasts to cover a certain part of our body that's not an implant?
Yeah, so that I would say is wound repair, for example. So whenever we get a deep wound, like a surgical scar, for example, or when the surgical wound heals, sometimes it leads to a scar. There is pathologies such as hypertrophic scars, keloids. These are basically big scarring diseases. And these scars involve, many of the same processes like inflammation and fibrotic processes, fibroblasts and such. And we know that these processes, this scar formation, is also activated by mechanical force. Sort of that was what the work of Dr. Jeff Gertner was, which inspired me to come here to Stanford, because a lot of the same cells and same mechanotransduction pathways that are involved in wound healing are also involved in biomedical device rejection. So now we're testing those concepts in the context of biomedical device, because, when you implant a biomedical device, you're basically creating a wound and then you're placing a biomedical device. So it is basically wound healing with the additional problem that there is now a device which is, mechanically very different from the rest of your body. So I would say this.
Of these cells in the body that attack an implant, you're saying you didn't realize that there's different types of cells that do it, or subsets of cells, and there's certain ones that seem to be a lot more active in the formation of fire. Okay.
Yeah.
So where are you headed with that path? Like, have you identified what kinds and why and their method of action?
Yeah, so this is, work in progress. We haven't published this, but basically we've found certain subsets of macrophages that are really active in this process, which populate the majority of these cells that are found on these capsules. And this we've found across different types of implants. So what we're seeing is that, one of the unique things is, again, the fact that all these devices are rejected, and all these, sooner or later, but all these devices elicit this foreign body reaction, and regardless of where you implant them. So this gives us a clue that there are some, conserved processes that are common across the foreign body reaction to different types of implants. So what my approach was to basically see what are the common cell types or subcell types that are associated with these capsules that are formed around these implants. And we found that a specific subset of macrophages, which have some elevated mechanotransduction pathways, are really very important, and we're seeing if we can target them to find therapy.
Can you talk a little bit more about mechanotransduction? What's happening? Are these cells sensing that the movement of an implant is makes it foreign, and I don't quite understand what mechanotransduction means. What the implication?
No, actually, that's a very good question, and it's a good question because mechanotransduction is a generic term we use. Mechanotransduction basically means it's the process by which mechanical cues get converted into biological, cues. So mechanical cues can be in the form of actual device movement, and people have shown there's some evidence to show that when the implant itself moves a lot, there is, accelerated rejection. There's also mechanical cues in the form of mechanical mismatch. You can imagine a pacemaker has very different mechanical, for example, stiffness than the surrounding tissue, right? So when there is a mechanical mismatch between the implant and the surrounding tissue, that again causes an adverse reaction. And there's also force itself, the tension of the skin, the force which the implant is under when these implants are put inside of the human body. So the force is obviously sensed by the cells. So mechanical cues in the form of force, device movement, as well as mechanical mismatch, all these can translate into adverse reactions in the body. So by targeting mechanotransduction pathways, in some ways, if we get to a central mechanotransduction pathway, we can sort of target all these mechanical cues at once. Does that clarify?
Yeah, okay.
Yeah.
So where do you think the big win is going to be? Is it going to be in the surface chemistry or the surface science of implants, or is it going to be in altering their shape, or is it going to be in targeting or suppressing certain immune cells or macrophages so they don't attack the implant? What do you think the win will be?
That is, again, a very good question. So I did my grad school work on, changing not surface chemistry, but surface nanoarchitecture. And that showed some good results. We were able to come up with certain nanoscale geometries of materials that would reduce foreign body reaction in the animals, of course. And now I'm trying it from a different angle. This is a clinical research program, so we get to work with these human samples. I think, we are getting at different subsets of macrophages and fibroblasts that are really driving this process. So I think the real answer is going to be a combination of things. I mean, bioengineering as a field itself is very interdisciplinary. As you might know, people come into bioengineering with different kinds of background. You can study materials science and then come into bioengineering. You can study, basic biology. You can study genetic engineering. You can study all these different kinds of sciences and come into bioengineering. And I think the final answer is going to be a combination of different things. Maybe it'll be design of different devices which can withstand more mechanical forces or distribute the mechanical forces. In addition, it'll be surface manipulation in terms of nanoarchitecture. And on top of that, it'll be, drugs targeted at mechanotransduction pathways. So I think it's going to be a combination of all these different things, which is why, again, we need different kinds of scientists working on this problem.
That makes sense. Any other things that happen in the body naturally that cause mechanical stressors? I mean, what about substantial weight gain or losing a limb?
Yeah, I think these are good hypotheses. Now, do we know if, there's a correlation between obesity and biomedical device rejection? Or do we know there is a significant correlation between aging or other kinds of diseases and biomedical device rejection? These are actually very good questions, and we are just about starting to answer these questions. And this is actually where data science comes in. We just about started this collaboration with Dr. Nigam Shah at Stanford. So where we're using data science methods to screen many medical device records and also look at the genetic and proteomics data that we're getting from our research to combine these and see what are the other risk factors that are associated with accelerated biomedical device rejection. And these are questions that we're beginning to answer. So I would say these are all possibilities, but we don't know them for sure. But now I think over the next 5-10 years, we're going to have answers because the tools are ready to answer these questions.
What do you see when I imagine like your lab in your room, you have like a big pile of pacemakers, so like, all kinds of devices, like a big junkyard in your room. I'm sure it's not like that, but what do you see when you get these devices after they've been taken out of somebody? Are you really surprised at how they look? Do they look mostly the same? Are they covered in thin films? Are they like completely crusted over like a coral in the ocean, or what do they look like?
Again, every device is different. So the way the process works is that first, I go and talk to the patient and ask for their consent if they are okay with it. And if they are okay with us collecting the implant, because, when these implants are taken out, they are basically dumped. And I take it instead of putting in the dumpster. I ask them if I can take it for research. And if they're okay with it, then, after the surgery, I collect the implants. And when I come, I basically get the implant and the little bit of tissue that's attached to the implant. Usually, it's a little bit bloody because of the surgery. But once the blood is washed off, the capsule itself is like this translucent white capsule that's on the implant. And that capsule is, mostly protein, mostly collagen 1 actually. And it's got obviously all these cells, macrophages and fibroblasts inside of the capsule and on surface of the implant. So every implant looks a little different. But what we try to do is then we take the capsule itself, digest it, take the cells out, and then, do what is known as single cell sequencing. So we basically sequence every cell that is there and get the sequence, so the genetic information in every cell, and see, what genes are activated. And then we also look at the surface of the implant itself under like scanning electron microscopy, for example. And there we can see a few different things. One, obviously, we can see the surface covered with the capsule. So collagen fibers are very nicely visible. And another peculiar thing that we observe is the formation of what we know as foreign body giant cells. So these are macrophages that fuse to form foreign body giant cells. And the presence of foreign body giant cells on implants, again, we've observed in mice and humans as well, is a sign that, the rejection process is underway.
You said that what kind of giant cells are created? Can you talk about those a little bit?
Sure. Yeah. So one of the peculiar things that, macrophages are involved in a variety of processes. Any inflammatory process, any wound, there'll be macrophages involved. But one of the unique things that macrophages do in the context of biomedical implants is that they start fusing with each other. And they fuse to form what we call foreign body giant cells. So basically, these are giant cells that are formed when there's a foreign body there. And these are basically, multinucleated macrophages that are pretty big. And these giant cells, they produce, biochemicals such as cytokines and chemokines, that again, they invite more fibroblasts, accelerate the rejection process, and so on. So they assist in the inflammatory process, and they also release biochemicals that attract more fibroblasts to come in. We know that foreign body giant cells are formed in some other chronic inflammatory diseases, but most often they're found in the context of biomedical implants. So this is a very unique thing where, when there's an implant, the macrophages start fusing and forming these giant cells. Now, why exactly this fusion process happens is still a matter of debate. We still are trying to understand, what is it about these what is it about these implants that starts this fusion process, and what is the exact role of these fused cells in the rejection process? Obviously, we have some idea.
Well, I figure it's like a self-assembling brick wall. They're probably more effective when they're all together in larger numbers. They could form a superstructure It's bigger than their individual cells, and that accomplishes their mission, right, of isolating the implant from the rest of the body. That's my guess, but
to some extent, yes. For example, so macrophages do this thing where it's called phagocytosis, where they, there's a piece of bacteria or some other pathogen that needs to, be gotten rid of. The macrophages can basically eat them up, right? So when people started finding, these giant cells on biomedical devices, the initial hypothesis was that this is a frustrated phagocytosis. So since they're not able to engulf the biomedical implant that's bigger, maybe they're trying to fuse to get bigger, and then they will sort of engulf the whole implant. But that hypothesis was disproven when people showed that phagocytosis and fusion are actually different processes. Another hypothesis is that it's like a defense mechanism against apoptosis. Apoptosis is, cell death, automatic cell death. It's because the cells are exposed to such different mechanical environments that they, want to apoptose. And one of the defense mechanisms is, as you said, they would create a more potent cell by fusing and becoming more potent. That is, again, a hypothesis that's not been proven to the full. One interesting fact that I would say, though, in my grad school research, what we found is that if we create, 55 nanometer nanorod arrays, we can actually stop this fusion process from happening. The cell macrophages don't fuse very well when there are certain nanoarchitectures on the surface. So why exactly does it happen? It's still a work in progress, but we know that it's very much involved in the foreign body reaction, and we know that to stop it is a good thing. But again, I think more research is needed to gain more clarity on it. The actual isolation of the device happens by the capsule that's formed. So the macrophages come in, they release, chemokines and cytokines that can activate other cells as well as damage the implant if it's made of polymers and such. And then the fibroblasts come in, and fibroblasts can produce a lot of collagen and other proteins. And then the final isolation that happens is because of this encapsulation layer, this proteinaceous encapsulation that happens. And that protein layer is what surrounds the implant and isolates it from the body.
what happens once you get a complete capsule? Does the body stop and say, we're done, we did our job, or does it keep going?
That's a very good question. Yeah, for the body, it's a defense mechanism, right? So if it's a way to keep it inside of a capsule, and that way the body now only sees collagen, which is its own protein, and doesn't bother it. Obviously, this is a very ideal situation, because even if there is capsule, there might be some release of some particulate matter from the implant and such. In some cases, I've read that when there's a bullet, for example, inside of the human body, and sometimes people, and if there's a nice capsule that is formed, sometimes people decide to leave it alone rather than go and operate and take it outside if it's like a reaction that has stopped after the capsule formation. But in many times, even after the capsule formation, the things could get triggered again, or if the capsule breaks loose and such.
Yeah, because I guess if you were able to create an implant with a surface that attracted collagen like crazy really quickly, maybe it would get to a stable state, and the body would stop attacking it. But it would be constructed in such a way that it would, maybe you can minimize the surface area of the implant that would be subject to attack very quickly by, again, growing collagen on top of it initially before it's implanted, or, having it happen quickly, and then maybe only a small area would be attacked, and it may slow the rejection of it.
Coatings of different kinds are actually, one of the ways people are trying to alter foreign body reaction, and it's shown some good results. But the one issue is, it depends on the application. For example, if you're building a biosensor that needs access to your interstitial fluid or to your blood, even a little bit of capsule, which stabilizes this process, is going to give you wrong readings. Because you can imagine, if the device has to access interstitial fluid through nothing versus through a layer of protein, there's going to be much less signal coming. So you're going to get lower readings, even though your glucose may be higher. So in those cases, it is not. But there are certain cases in which, in breast implants, a small layer of these capsules can actually help stabilize the implant. But again, it depends on, what the specific application is and how well you can control this reaction. One of the sort of key areas of thinking is that it's immunomodulation that we're going after, not immunosuppression. So you don't want to stop the process completely, but you want it to get to a stable state where the implant can still serve its function and at the same time not elicit more and more of this reaction as we go forward.
Makes sense. Okay. So how far away is your research from being able to being used in implants and in implant situations? Are you just really on the hardcore research side of it, or are you working towards a product solution?
One of the reasons why I came to Stanford and also the Department of Surgery with Jeff Gertner from a bioengineering background was because of this ability to translate things from the basic science to the clinical science. The research that we're doing is, of course, based on animals, but also these human implants that I talked about. So once we have this and we can develop some drugs and we can show that it works in mice, we can, go for large animal models and start translating them. So currently it is still at the research level, but I think once the technology works, the sort of process to translate this into humans is within a few years, and we have the expertise here. And Dr. Jeff Gertner, specializes in translating some of these technologies to the clinic. He's done some of this work with wound repair technologies, and I believe we can do the same with biomedical devices as well. It's just a matter of finishing the research and doing the due diligence before we start thinking of translating it.
Yeah. All right, well, that's great. Jagan, thanks for coming on the podcast. And if people are interested in getting in touch, what's the best way for them to reach you?
The best way is my email. It's Jaganpa at Stanford, JAGANPA at stanford.edu. I also run a blog called sciencers.org, which people can reach out to me. I think those two would be the best ways to reach out to me.
Very good. Well, Jagan, thanks for coming on, and it's very interesting work that you're working on.
Oh, thank you very much. Just one more thing, Richard. Yeah, it's you asked me the average lifespan of devices, and for breast implants, I think I said 7 years. It's about, the average answer is about 10 years. So.
Okay, so a breast implant will last how long in the body on average?
The average is about 10 years, but of course there's less and more depending on.
Right.
Yeah.
Okay.
You have been listening to Almost Here, Around the Corner Future Technology podcast with Richard Jacobs. Subscribe to this podcast, post a review to discover more future technologies that are poised to transform our lives for better or worse, such as Bitcoin, artificial intelligence, 3D printing, blockchain, virtual reality, and more.